Here we go. Hey everybody, welcome to the Growing with Fishes podcast. We have a very special episode for you today, episode 203. We have Mr. Kevin McKernan joining us again for a second time, talking about his new paper on COVID-19 and cannabis testing. He's got some really cool stuff to talk to us about some advancements with some of the different testing methods he's been doing to test some stuff around the cannabis industry and some of the components and inputs. So it's going to be really interesting to talk about and to learn what he's been working on. Thanks a lot for joining us, Kevin.
Thank you for having me. Pleasure being here. I'd love to share what we're up to in this COVID nightmare we're all running through. So hopefully everyone's safe on your end and it's not having too much disruption in your life.
Oh, thankfully, I'm on a farm in the absolute middle of nowhere, Oklahoma. So we do a pretty good natural social distancing without having to try. So it worked out well. We have plenty of food supply here as well, so no worries. We also have Roger from I Love Growing Marijuana.
How's everybody doing tonight? I think you're going to really enjoy this new information that we're going to share tonight. So enjoy the show.
And we also have Marty.
Hey, what's up, everybody?
AP Meds. And then you have myself, Steve at Ponics, and you can also find me at True Aquaponics if you're looking for nutrient subscriptions for your aquaponic system or aquaponic nutrients. And then you can find the podcast on your favorite podcast app or on YouTube at Potent Ponics. So thanks a lot, Kevin, for joining us. So tell us all about your new paper. You've been working very hard on a lot of research around COVID-19 and cannabis. Please tell us all about your research and what you've been up to lately.
Sure. So I guess as just a primer to this, I've done a lot of work digging into the ancestry of the virus as well, and I'll point people to a few other YouTube videos that discuss that. There's some interesting details in this virus. I actually don't think it's as frightening as the news is making that out to be, but that's probably doesn't bear much repeating. It's probably obvious to a lot of people. Yeah, there are certainly people getting sick, and this is a concern, but it's very much a concern for a specialized group of people. And I think that's very much true in a lot of cannabis testing, is we're really looking out for the immunocompromised, and many of the things we're putting in place are probably superfluous for a healthy individual. So we got involved in looking at this, somewhat dragging our feet in that we didn't really think COVID was going to be around long. We thought it was going to kind of buzz through. I still think the second wave is probably going to be very weak, if at all. But I do think it's seasonal and it may be back in a different form next year. But that being said, there were some people in Massachusetts that were getting sick at the grows, and then people didn't know what to do with any of the material they handled. How long does it last on cannabis? Does it replicate on cannabis? Is this thing a vector? Is it a fomite? Fomite's a term for something. It's a fancy term for what the virus might land on and then move it to somebody else, but not allow the virus to replicate on it. So, and I think that's what we have here with cannabis, is it's not a replicative host. There's no data for that, but it could potentially carry virus to another individual who might inhale it. Now, I don't think, the pyrolytics in most flames are probably going to kill this thing, but there are some people vaporizing at lower temperatures where we don't know if that's the case. Most of the work on sterilizing for SARS-CoV-2 is using about 65 C for 30 minutes. Not a very high temperature, but a long time. And what we don't have is what does 130 or 150 C look like for a few seconds. That's information we still don't have in the cannabis field, and our paper doesn't address that. And there are some challenges to addressing that because it's a bio-level 3 pathogen, and it's very hard to get your hands on live virus of this right now. And I don't think the CDC is going to be handing it over to cannabis testing labs anytime soon. So yeah, we started making a test for this just to pick up the RNA on the plant, and it really came out of the virus team at MGC, who's been working on hops latent viroid and looking at lettuce sclerosis virus. These are plant viruses that do replicate in the plant, but they're also RNA viruses, which is very similar to the SARS virus in terms of that it's an RNA virus. It's probably the only similarity. Can you hear me still?
Yep. absolutely.
Yeah, so these RNA viruses are kind of in our wheelhouse, and so we decided to try and expand this and test this on cannabis to see, can the plant harbor it, or is it going to kill the thing in like 24 hours? There are some surfaces that were published that SARS-CoV-2 just doesn't last very long on copper surfaces, for example, but lasts like 4 days on plastic surfaces, and we're storing a lot of our cannabis in plastic right now, so the plastic is just as much of a fomite as the plant. Excuse me. So we started building tests for this so that we could test a gram of cannabis. And it's a little bit trickier to do testing on cannabis than it is to do it on the nasal swabs. Has anyone here been tested for COVID yet? If you're way out in the woods, you probably aren't going to encounter it. But
Yeah.
it's a fairly unpleasant test where they shove a nasal pharyngeal swab up into your brain or into your cavity. And not quite your brain, but it feels like your brain. And they collect as much, cellular debris as they can on that swab, pull it out, dip it into a 1 ml of fluid, and then they end up PCRing a 1/250th of that fluid. So they can't detect anything less than 250 copies of the virus that's on the swab. We had to get down to those sensitivities, trying to pick out something out of a 1 gram cannabis sample that's in 15 mLs of fluid. So it's 15 times harder in essence to get this done on cannabis. So we've got that now worked out. And that's what the preprint demonstrates, is that we know we can detect this. We know how low we can detect it. We also know how long it can last on the plant. And we did time studies out to about 7 days that this can last on the plant. But I want to be really clear. We've never found this on the plant. This is not something that we think is on cannabis right now, but it could be put there by human contact or potentially by bat guano. And that's something that's probably worth mentioning to your audience, because I know your audience is quite aware of the benefits of using a lot of these other natural fertilizers. They're very important. I really believe in getting these organics to work and getting a lot of the beneficial microbes out of them. But the main coronavirus discovery that's ongoing right now, they're finding most of it in bat guano. Now, these aren't SARS-CoV-2. These are its ancestor, rat G13 usually, or some other derivatives. So we don't know if they can make zoonotic leaps, but that's where people go if they want to find new coronaviruses. They sequence bat guano. So that's one thing we ought to be aware of if we're fertilizing. And now, I've not seen any bat guano in the United States test positive. I don't even know if anyone's ever looked. They're really finding this stuff out in the depths of China. And so it may not even be circulating in bats in our country. That's something that yet to be demonstrated. So I guess just know your source if you're sourcing that stuff as to where it's coming from. And there are now some tools you might be able to use to test for it to see whether or not it's in fact a risk. So that's where we started. I don't know. I don't want to monologue here on you. So I thought I'd at least throw it out for some questions.
No, that's very interesting. So tell us more about the potential for someone to contaminate, maybe a dispensary employee, or in some states they allow people to smell those 1 pound jars, and
oh god, I didn't think
someone
about that.
cough or sneeze into that and have that, and that could end up falling back onto the liability of a dispensary, and they had no idea. You know what I mean?
Yeah.
So what is the actual real world potential for that to happen based on the test results and that you found?
Well, so the interesting thing is there are some intersections here with the cannabis field that we got to pay attention to. So if you actually look at currently SARS-CoV-2, this thing does not have, it's not necessarily lethal to a healthy person. Most of the deaths that we're seeing, probably over 95% of them, I think even in Italy it was closer to 98 or 99%, are comorbid patients. These are patients that have either diabetes, hypertension, COPD, or cancer. So they're immunocompromised in some way, and the average age is like over 80. So it's a much older, vulnerable population that's getting these. However, as you know, in the cannabis field, cancer and COPD are commonly used. Now, I don't think people are inhaling it necessarily for COPD. They're probably using oils, but in cancer, there is some inhalation that goes on for people who are just trying to get quick access to pain meds. And they're dealing with cachexia and nausea, and they can't always take things orally because they throw them up and don't know what their dose is. So there is a lot of inhalation that is utilized in cancer palliative care. So that's a concern, that if we've got a population that in fact has similar or an overlap of the patients that might be users of cannabis also happen to be comorbid with these things, and that's one flag to keep your eye on. The second thing that that we're seeing with this, as we mentioned, there's fertilizers and there's people who are potentially human contact that could be going on here, but there's also this potential risk that we're not properly drying or curing this, or potentially sterilizing the plant before somebody uses it. And so I think if we get into the extraction space, this isn't necessarily a concern. I think when you start getting into vaped flower, we have to start thinking carefully about it. I think that's probably the biggest vulnerability. That's where we have seen aspergillus take hold in some immunocompromised patients. There's a paper out there from someone by the name of Remington. And if you Google Remington et al. And aspergillosis, you'll find case studies of people who are dying from getting aspergillus into their lungs. And that's the one paper that did the responsible thing and actually sequenced the biopsy in the patient and sequenced the microbes in the vaporizer and found a match. Usually that's never done. It's usually associative. And I think that's an important thing to remember, because as you're going to see as I walk into this COVID story, microbes are very ubiquitous and they come out when your immune system's weak, but you might actually be harboring them all along. And I think that's true in the case of some viruses as well. So when you look at the patients that are dying in COVID, 50% of them die with a microbial infection, a secondary infection. Aspergillus is one of those. Candida is one of those. There's a list of these things that when these patients get this virus, their immune system gets weak and other opportunistic pathogens take over. And so some of the complications are actually from the other pathogens, not necessarily from SARS, because usually by the time, sometimes your immune system is actually doing more harm than the actual virus. It's called a cytokine storm. So there's a lot of dynamics going on here, but we've got immunocompromised patients in the cannabis field. We have an inhaled product, and this is something that you're starting to see places like China institute testing of all the meat now. They just announced today they're going to be screening all the meat, only incoming though, which is weird. That might be political, but they announced imported meat will be screened for SARS-CoV-2. So our biggest fear is we just got off of this Valley thing. And I don't know if you guys followed that whole vape crisis, but if you look at some of the symptoms of Valley, ground glass opacities in the lungs is one of them. And that's shared with COVID as well. A lot of these pens came from China. This predated this pandemic and just kind of petered out of the news as this thing came to play. Now, no one found Valley to be contagious. I don't know if anyone really looked and considered that. And it might be a very hard thing to detect if there's a lot of asymptomatic spread with COVID. That's a point that's in debate right now in the scientific field. How much is being spread asymptomatically? The WHO is flip-flopping on this. I think a lot of the research is kind of still being thrown about as to how much spread of the virus is happening through the asymptomatic population, but it's in debate if it's very high. What is known is that it's unlikely low exposure levels to the virus or what get you sick. Is you need to be in one of these parties or in like a super spreader event where you get exposed and hit with very high viral load. And that's what really kicks off the infection. So a lot of dynamics here to think about. I'm somewhat suspect that a cannabis plant, unless someone directly coughed on it, will get enough viral load that would end up in someone's lungs, a week later. But those are things that we need to be able to quantitate. We have some of the basic tools now to begin to do that. But there's one hole in what we can do without a BL3 laboratory is we can't tell you the viability of these viruses without propagating them. And I don't want to get in the business of propagating this virus. So we've just been measuring the DNA levels and the RNA levels, I should say, of this virus. And that's something we can do. We've built some assays that do some like live dead assessments of this through enzymes, but we don't have the rock solid evidence of this thing growing as it comes off the plant. We just can see the RNAs there. That's the same thing that the PCR tests are doing for patients as well. And it's one of the reasons why patients continue to PCR positive for a long period after their symptoms subside is we think that's just lytic virus. It's not necessarily contagious. It's just your body shedding the virus. But we don't know. We don't have those studies done yet because this thing needs to all be done in BO3 laboratories. So yeah, that's kind of the backdrop of what's going on. Is there are some unique aspects of the cannabis field, this being an inhaled product, there being fertilizers and people that have come down with it that are handling the plant very manually, and there being comorbids in the population that intersect with the comorbidities that we see in COVID. So those are really the reasons why we kind of said, all right, we got to take it a little more seriously than just brushing this aside and hoping it goes away. That our main fear, you're in a different state, but during a valley, Charlie Baker just shut crap down here. A bunch of vape shops just went out of business. He's just closed them down, made it illegal to do any flavored vapes. And I think most of the vape pens disappeared off the market and everyone went back to flower. And flower became extraordinarily scarce in Massachusetts for a period of time. So I think a lot of people are still utilizing flower now because I don't think the vape pens have really totally resolved in people's collective comfort level. So there still is a lot more flower usage going on. But we get one wind of this getting into the cannabis field, and if they can shut down the NFL, they can shut us down.
Oh yeah, we're starting to see solventless come back as well. Solventless production seems to be going up in a lot of places as well, where the vapes have kind of slowly died back. So it's been interesting.
Oh, interesting. Yeah. More water hash, bubble bags.
People are educating themselves on what they were actually intaking, and then realizing they still want to get high, but they want that clean product now. So
yeah.
So tell us more about this bat guano vector with the plants. I know I've done quite a bit of study, and there's been excuse me, quite a bit of debate on the side with
dry coffee out there, Steve.
Oh yes, COVID cough. No, it's a dab cough. We actually got some nice new check. If you're in Oklahoma, we have some Aquapunch.
Oh, nice.
Really awesome. We have these giant, and yes, this is one giant diamond.
Wow, THC diamond.
So all live diamonds in sauce. It smells amazing, tastes amazing, and is smooth as glass. So definitely look for that in your stores out there in Oklahoma. But so tell us more about this bat guano vector, and so I've done a lot of study with plant pathogens, especially in aquaponics. There's been a heavy amount of debate on whether or not you can actually transfer a lot of pathogens. University of Hawaii actually did 7 years of study on that specific problem, on what types of pathogens they intentionally introduced pathogens, and then just tissue sample study in both fish and plant tissue, as well as fruit tissue, leaf tissue, stem tissue, and all that stuff. And they weren't able to find very many pathogens, and you had to get to kind of psychotic load levels that were well beyond any kind of normal commercial, anything you would ever sanely do in a commercial farm. Before it was actually transferring through, because plants do have some level of filtration at that root system, and then with the microbials that also help act as a defense. So what are you finding as far as the coronaviruses as a general group with the plants and the potential for them to maybe be uptaken through that? Or is it more about the direct contact with the plant tissue and then the secondary contact?
I think it's the latter. I would not. This virus wants an ACE2 receptor, which is nowhere in the cannabis genome, and I don't think it's going to find any entry into the plant. I mean, to be honest, I haven't looked very deeply. I kind of blankly wrote that off in my head that it's probably not risk. I think it's more the fomite risk that someone coughs on this thing or sticks into their nose while they're smelling it, and you get enough viral particle in there to potentially transfer to another person. I was somewhat shocked the RNA lasted 7 days, and it only went down about 150-fold across 7 days. That surprised me, because a lot of the other studies, granted, they were being done with viability on Vero cells. They saw a drop that was much faster than that in 4 days. But they're measuring it differently than we are, and their system is probably more reflective of risk than ours, because they're looking at if they can form plaques on monkey kidney cells. So that's but you know, it was our first try at this. We just had to just figure out, you know, look at how long this thing lasts. What is there any potential risk at all? If it disappeared in a day, it seems like there'd be a non-issue, but it's there a little bit longer than that at the moment. So, you know, in terms of the aquaponics, you know, one thing that was, I don't think we've spoken since this. There's another paper we put out in January where we did a lot of sequencing, 42 different cannabis genomes, some that were powdery mildew resistant, some that weren't. And started combing through the genome for the genes that are involved in resistance, pathogen resistance. And we found a few that we cloned and expressed in E. coli and started studying. But but one's a chitinase and one's a thaumatin-like protein. And the chitinase is most heavily expressed in the roots. So this is something that dissolves fungal cell membranes by dissolving chitin. And the other one is a TLP, or thaumatin-like protein, dissolves beta-glucan. So those are two of the really key features of a cell membrane that these things dissolve. And we can put those things in culture with Aspergillus. And those enzymes, now that we've expressed them out of the coli and have big vats of them, we can put them into TSB that we use to grow Aspergillus, to do typical Aspergillus testing. And it whacks the growth down 100-fold to 1,000-fold. So they do have a real impact and do fight off Aspergillus. And it's really interesting that the roots are the things that are that have none of these things expressed, because the roots, I think, are trying to communicate with microbes, particularly mycorrhizal fungi. Whereas the rest of the plant, the most heavily expressed gene in Jamaican lion is a chitinase, more than THC synthase. So it's a heavily expressed gene, and one of the I think key things for interacting with a lot of the microbiome.
It's really interesting. When I was in Africa, we were having problems with grasshoppers and stuff like that, and we started doing are you familiar with the Korean natural farming?
I'm sorry, did you say what kind of natural farming?
Korean natural farming, KNF, with the IMO collection, with the rice collection of local fungi, saprophytic fungi.
No, I'd love to learn more about that.
Oh yeah, you take rice and you cook it most of the way, and then you put it out in a box and you collect the fungi, the mycorrhizal fungi with that, and then you can culture that with sugar, mix it 50% with sugar to stabilize it, and then you can, there's other steps you can do to further process it as well. So what we were doing there was taking that, replacing about a third of it with insect frass, mixing that with the rice, and then doing collection for chitan feeding microbes that were directly feeding on a lot of the pests that were attacking the plants, and having a good one. But I wonder if that also, because they were directly feeding on the chitan, I wonder if the byproduct was making it more available for the plants as well, because it seemed like it also had a noticeable effect on any molds and mildews we had as well, even though it was mainly focused on feeding on the insects, at least as far as the insect frass being the source that we were using to collect it on. So
the insects all have chitin shells.
Yeah, so I was curious if that had played in, maybe you have more thought into that, or maybe it's the liquefied chitinase from the lactobacillus mixed with the lactobacillus, but the chitinase with the sugar in a liquefied form. Maybe that's the actual thing that was increasing the plant's resistance on the mold.
It's a good question. I mean, certainly those enzymes do break these polymers down into monomer units, right, into smaller, and those might be more digestible carbon sources for other microbes. So there could be a some of that going on. I mean, that's effectively like malting. It's very similar to malting and making a mash and brewing, is to break down some of these carbohydrates into smaller subunits so the yeast can actually ferment them. In fact, thaumatin-like proteins are often used for that. I mean, the assay that we have, interesting enough, one of the assays came out of the brewing industry. To measure the progress of your mash, you do what's known as a beta-glucanase assay. Really easy, simple to do the spectrophotometer. I know I don't. That's a good question. I don't have as much background on like insect interactions. I mean, we've done some work sequencing, russet mites. We've sequenced the russet mite genome, and we didn't even really look at it or study it other than to make a PCR marker, just so we could have a test for it. It's really sad, David Fair's, that you sequence someone's genome just to get a PCR test out of it. But we can do that now. It's really cheap. And God, I think the spider mite's got to be next. I think that one's actually been sequenced, And I believe it's the same one that's on cannabis that's on other plants. I think the Broad Institute may have sequenced the spider mite genome.
The two-spotted spider mite. Yep. And then
Yeah,
there's the
that thing's a pain in the
mite.
ass. So we got to, hopefully that leads to something. But yeah, no, our efforts have been looking at what we want to do is get genes that are in some cannabis plants that are missing in others, and express the enzymes for those so that they can be used as supplemental, like supplemental enzymes. So Jamaican lion's a powdery mildew resistant plant, and its powdery mildew resistance, we believe is maybe it doesn't explain powdery mildew resistance in all plants. Let's put it that way, because we've gone and sequenced a bunch of other people's plants, and they have a different collection of these genes. There's like 80 of these genes, and so so there's probably convergent evolution going on here, where there's multiple different ways where plants can become pathogen resistant, and we just happen to characterize what's going on in Jamaican lion. But the enzymes, the two enzymes that the lead candidates for all the activity, we pulled out, put them in E. coli, expressed boatloads of them, and then put them back into culture, and we can see that they're functional. Now the question is it economical to utilize this as a spray when everyone can just do sulfur burns or use hydrogen peroxide or something? It may not be as economical. We don't know those numbers yet. We have to dive into that. But
the downside is when you do sulfur burns, it makes your hash taste like matches.
Yeah, right. Yeah, that's I don't know. What about the other treatments out there? I mean, what do you guys use?
So actually, for powdery mildew, I've found nothing works better than lactobacillus, hands down.
Yeah, there you go.
But then a rotation of Bacillus subtilis, Bacillus pumilus.
Those things probably make kinases.
On rotation with labs works awesome. And then the other thing that I recently learned, and this was per advice of breeder Steve, is cooking the greenhouse. He goes, in Colombia, we can't import all that crap. He goes, we just cook the greenhouses. He goes, powdery mildew can't survive above 95 degrees Fahrenheit very long. So you just run it above 95 degrees Fahrenheit for 1 hour a day for 7 days in a row. And, it's very hard for that to live. So, it's something that we're experimenting with here in Oklahoma as a prevention. But I'll tell you what, we started running it 3 times a week above 95 degrees just for an hour. And it is, I've found 3 leaves in over a month, and they were on some, 1 week plant. It was on one single plant, and that was it. Wow. Over the course of that whole time. And that's in a greenhouse, an aquaponic greenhouse with all this high humidity and basically perfect conditions if you're going to actually predict where to grow it. And we don't have any problems. You know what I mean? But we're doing probiotic sprays and good ventilation and good air movement, and we don't have problems.
Well, that's going to be better than the labs are great, lactobacillus, but you do run the risk, depends on what state you're in, you run the risk of failing total aerobic counts with those.
In flower, yes, I would never use it in flower, but I'm talking veg and clone production and mom production.
I see, early on, they by the time.
In flower, you're stuck. And this is where it gets tricky, especially if you're trying to do something organic. After week 4, you really can't use Bacillus pumilus or Bacillus subtilis and still pass colony forming unit testing if they're going to do bacterials. So they'll grow CFUs because they just put it on an agar. They just roll an agar plate.
And they don't care what's on there. They're just they're just blindly counting colonies.
Exactly. So. And there's all different types of ways to make sure your sample is sterile before you send it in, which you can decide if you need to do that or not based on your methodology. But the best, what you really need to do is educate your regulators on being able to test for different pathogens and not just doing these plate
we're working hard on that, and I think we're making some headway in a few states where
and you provide some of the best hardware. And you provide some of the best hardware for people to be able to do that. So do you want to tell us a little about the qPCR and stuff for those of you that maybe aren't familiar with your stuff and maybe
sure. Sorry about that. So we make a lot of the test reagents for testing for Aspergillus, E. coli, Salmonella, what have you. We have an arrangement with Agilent to we now have instrument deals, so we can help you guys get the instruments and the reagents. These are quantitative PCR instruments. This is the tool that's being used for COVID testing. All 100% of COVID testing right now is on quantitative PCR platforms because it's just the most sensitive tool out there for diagnostic testing when medical issues are on the line. So That aspergillus assay right now is out. It's just getting its. We've had one out there that tests for Flavus fumigatus, Niger, and Terrius. We just updated that one to be a 5-color assay, so it speciates these things on the fly. In the past, if it triggered, you didn't know which one of the microbes it was. You just knew you had aspergillus. Now some people are asking, can you speciate it? And so we've turned that into a multicolor test. We also make some PCR tests for sex testing and for testing for THC, CBD, genetics in the plant. The paper we published back in January, you can discern the class of plant that you're going to have genetically. We can't yet tell you whether you're going to break 0.3. We're getting closer on that. But we can tell you if you're going to be a type 1 plant, a type 2, type 3, or a type 4 plant just from the genetics alone. Where we're putting a little more effort right now is tracking the cannabichromene genes, because those seem to be another source of potential leaks. I think CBD synthase is the largest leaker we've got out there. So if you're making type 3 plants, it's going to always leak some THC on you because of that gene. It's hard to get rid of that unless you go to a CBG plant that doesn't have one of those genes, in which case you can get the ratio even better. However, what's leaking on the seed, there is still residual amounts of THC on the CBG plants. And we think those are probably coming from the cannabichromene genes that are in some portion of the population. There are a large host of plants in the population that don't have a cannabichromene cluster. It's like a 2 megabase deletion in the plant, and some plants don't have that. So interesting enough, on that same 2 megabase stretch, there's like 4 or 5 other genes that are involved in pathogen response. So when that cannabichromene gene is deleted, you're losing some aquaporin receptors and a couple other things you got to keep your eye on. So we make a lot of these genetic tools that test for this. That's the Agilent urea that does 96-well quantitative PCR. We were also rolling out a SNP chip right now that does 90,000 SNPs. This is getting done. This is done on an Illumina platform, but it's being run through Eurofins. Eurofins is a network of labs all over the world. They have some great labs up in Canada, and I think there's one in Longmont, Colorado. But this will test for genetic diversity. It will test whether the plant's male or female. It will give you the type 1, 2, or 3 status, and then a host of like 50,000 different coding variants in the plant and some evenly spaced markers throughout the genome. We've got the flowering genes on there. We've got the canflavin pathway is in there, which is something that we're really interested in, and there's some ties to COVID to that. Hopefully we'll touch on. And a host of other genes that people have asked us to track for them are all printed onto that chip. So those are just going through validation right now. The data's in Illumina's hands. They're doing some clustering analysis on this, and we'll probably have some of the first data this week on the first 100 or 200 samples that are run through that. So a lot of tools that you could use to do accelerated breeding.
Awesome. Really appreciate all the hard work you're doing. I wanted to circle back to something you had mentioned earlier that I had wrote in a note for it, and we had gotten off, and I wanted to circle back. So you've actually worked with lettuce chlorosis virus, and I've actually had the displeasure of seeing that twice when aquaponic facilities convert from large-scale DWC lettuce production with tons and tons and tons of seeds all the time, co-grow with cannabis. And it contaminates them, not paying attention either with pruners or an insect biting one and the other, or the exact thing we don't know, or if it's the water. We've also had some suspicion as whether or not it's actually water transferable for some short period of time. We don't actually know for sure, but the fact that they're directly next to each other, sure as heck makes it easy.
Yeah.
So I've seen that twice now in facilities that we're converting over, and you're probably one of the only other people that has actually done any amount of work or even seen it. So please tell us more about the work that you've done with it and the testing stuff, and what you've learned.
Yeah, so here's the thing about RNA viruses are really difficult to get your hands on. For one, anyone who thinks they have one of these things isn't really receptive to you asking them to save it for you so you can come poke around with it. So we've had a really hard time actually getting samples of these that are known confirmed viruses. So what we do in those scenarios, it's a little bit like SARS, is we design the RNA targets for these things, and we practice making our tests work on these synthetic targets. And until someone actually sends us material that has virus on it, which by the way, doesn't go across state lines very easily right now. So we're kind of relegated to either shipping our kits to you or to collaborators to try and work this out in the field where it's happening, or folks in the field being able to do RNA purification and send us the RNA. The latter one's a little tricky. You got to have some experience handling, doing pipetting and handling RNA. So at the moment, we're reaching out and placing some test kits in a variety of labs out there to look for these viruses a little bit more remotely to where they're happening in California and other places. But we can tune and calibrate all of these tests by feeding it synthetically, synthesized RNA that's identical to the RNA of those viruses, so that we know the sensitivity of our test. I think where the questions come into play right now are, how many times do you have to sample the plant? Because I doubt a single hole punch of this thing is going to nail it. You're going to probably have to sample it 5 or 6 times. We might need some swabbing techniques to cover the surface and get a little bit more concentration of the surface area into the test. Those are the things we're trying to work out right now, is how many tests you got to run to know a plant's clean. We don't know that number right now.
And you ran into some weird stuff with that too when you were doing the hop latent stuff, or with the hop latent stuff. No, maybe it wasn't you, but I know when they were doing the hop latent testing, they test one side of the plant and it has it, and the other side doesn't. And,
oh yeah, that's probably Jeremy's work up at Dark Heart. Yeah,
the Dark Heart.
They're actually pioneered the space. We're really following them on this. They're the ones who found it, and we just decided to make some tests that were multicolor that some folks can try and use. There's another group in Israel that found the lettuce sclerosis stuff as well, and we've just been working with their material. So we're standing on the shoulders of those giants on this study. But so yeah, last I spoke to him, he was sampling it like 4 or 5 times to try and find this, and it's not clear. I spoke to him as CanMed, so I haven't seen him in a bit, but his advice was, you can't get away with just a single sampling. So we're looking at some techniques where we can take 10 samplings and pool them into one test, so you don't have to do 10 times the number of tests to get this done. And that's kind of a study that's ongoing right now. Is can we pool multiple samplings into a single vial and then and ultimately concentrate that material and test more efficiently. So yeah, I'm kind of interested to see where those go, because those papers are both very interesting. But I do think that sometimes the phenotypes these viruses present on the plant aren't that clear. I mean, we've had a lot of people. So prior to us having these 2 viruses, we had other tests for something known as Cannabis cryptic virus and hops, and another hops virus, that HMLV or something, right? So, and people would send us in things they swore had viruses. They always thought it was tobacco mosaic virus, and we'd run these tests on them and we'd never get hits. So I think there's a lot of pleiotropy, if you will, of phenotype that you get. What looks like one disease can actually be multiple different diseases if you don't molecularly characterize.
Well, especially with mosaic. I mean, through Israel, I know there was a paper on lettuce sclerosis virus, cannabis, published in the last 2 years, and they cited a whole bunch of mosaic virus studies from an Israeli group. I believe it was with 12 or 14 different mosaic viruses, and it was Arabis, lavender, alfalfa mosaic, cucumber mosaic, tobacco mosaic, tobacco streak virus, hemp mosaic virus. They've documented a whole wide range of them now, and it's like, how many of the different mosaic viruses can transfer to cannabis? Just like the beet leaf curl virus and some of the others, it's like, how many of that group can transfer? And how, there's a lot of these are groups of viruses that are now being documented in cannabis tissue, and it's like, how many of that family can jump to cannabis, which is starts to get scary.
So here's the thing with this is I suspect in the next few years, we're going to see an explosion of discovery in viruses, because, part of the reason this COVID crap is happening is that our viruses are harder to sequence than bacteria and fungi. They're smaller, but bacteria and fungi, it's really easy to know that they all have at least a ribosomal operon that you can amplify, right? There's universal regions that exist in microbes and fungi that you can amplify and sequence them. There's good tools for fishing for these things. The viruses, though, there isn't like a universal sequence that you can use to go and amplify and hunt for these things. So you have to do very laborious next generation sequencing and RNA sequencing experiments to sort these out. And you got to be really careful harvesting it off the plant. You got to make sure that you don't get any RNases involved when you're doing this. So, hunting for these different viruses, there's just a lot more work in doing it. But now we're primed with the information that we need to go look, right? We're starting to get fruit from doing this, in multiple different labs, virus quickly once they point the gun there. And now that I think that pace has been set, I think more and more people are going to turn to, okay, I think I have a virus. Let's just give it a shot. Let's send it off for some next gen sequencing and see if we can pick it up. The bacteria and fungi are far easier. If you have any question on that, we can readily amplify those a single PCR reaction and get it in a sequencer and know to tell what it is. But the viruses really require a lot more care and to go after. It's odd considering how small they are. But I think the same thing is true in COVID, that there are a lot more of these coronaviruses are finding circulating, and they've been around. This thing has ancestors. They just weren't as symptomatic, so we ignored them and no one went hunting for them. But now we're finding there's patients that have prior coronaviruses that are protecting them from this one. So that means the immune repertoire that we have, the antibodies out there throughout the human population, it's not correct to call this thing a novel virus anymore, because it's not going to be hitting an immunonaive population. It's going to be spreading through the population with the brakes on, because it's going to bump into a lot of people that already have immunity. And that was never anticipated in the models that created this economic shutdown. That was kind of ignored, or just, I think there's a publication bias. You find something that hasn't been in the database before and you scream eureka, it's novel, and it gets broadcasted pretty heavily through the peer review system that way. When reality is it probably wasn't that novel, we just haven't looked in other places. And the way in which this virus is jumping around between mammals, like you're mentioning these viruses that may be jumping around between plants, we're seeing that now. We've seen SARS in minks. There's a bunch of minks that are being euthanized this week over in Europe, unfortunately. But minks have it, cats have it, ferrets have it, tigers found it in tigers. They've found it in earlier versions. They found in camels, but not this one. So it's in pangolins, it's in bats. So this thing seems to bounce around between anything that's mammalian. And they've even documented it going into minks and coming back out and infecting other humans. So I'm somewhat suspect that promiscuity is new. I think it's probably been happening for a really long time throughout this virus's history. It just didn't create a fuss, and we weren't paying so much close attention to it. At least that's what the antibody data seems to imply. So I do think we need to think of these viruses as things that can hop between plants. The ones that are in mammals are hopping. There is good reason to believe the tobacco mosaic virus may in fact hop, but it just may be mutated enough that people are calling it hops mosaic virus. And that's probably the more likely recent ancestor in hops than tobacco. That might be true for these lettuce sclerosis viruses and the viroids. The viroids are even more interesting. Those things don't even have protein coats on them. I'm just stunned that they propagate and survive.
Yeah, but isn't it true that most of the viroids have to be directly transferred through contact, and they're not?
Yeah,
really...
they have no dispersal mechanism. Yeah.
So it has to have an insect bite or dirty tools or some kind of other direct contact.
Yeah. And it's a really good question about the mosaics. I mean, are the mosaics require the same thing, or do they think those things somehow get?
So okay. Through my experience, I have witnessed, in fact, I have really good pictures of a grow where you can see the strain that had it, and in the mom room, I could see the mom that it came in on. This mom very clearly has it throughout the whole plant, and scattered throughout the leaves, and then the clones have it. And then you could see in the secondary veg room where someone had not cleaned their pruners right at a grow I was consulting on. And then the other plants down the row, other strains had it, whereas in the mom room and in the other rooms, they weren't expressing it at all. So as best that I could tell from the fact that this had been going on for weeks or months and months and months, is that they were trimming it and not cleaning their tool, the pruners, and then transferring it over, because I was only seeing it in those strains after they were moved on and not in the moms ever. So it wasn't because in the moms, each mom had its own tools, its own pruner, because of the way this place was laid out, whereas they didn't have that same situation in the veg room, obviously, because they have more plants. So that was just the way that they had. Maybe it wasn't even every plant, but they had it for every strain, right? So each strain had its own set of tools in order to prevent any kind of issues. Well, that was how I was able, one of the ways I was able to determine it. But the fact that you could just see it blatantly down the line and then see the progression of infection based on how far it was in the plant. It's, at least to me, visually seemed pretty obvious. And I've seen a whole bunch of that where you could see it in maybe in one or two strains, and then by the time it makes it into flower, you can see it into a whole bunch because people aren't being clean.
Yeah, well, I mean, there you go. I mean, you're cutting right into the plant and going through a lot of its defenses, getting right to the vasculature. I mean, can you imagine if we would trim fingers on people during COVID, like how quickly it spread? we weren't like changing the scissors. Yeah, that's a prime way to make these things move.
So what types of viruses if at all, do you have the ability to test for with your stuff currently, or is that something you're
right now we have a test that uses an RT-qPCR. That's a reverse transcriptase PCR that goes after hops latent viroid, lettuce sclerosis virus, and I think cannabis cryptic virus is the other one that's in there. Granted, we know how to run these and target positive controls that are in the kits. Where we're looking to collaborate with people on is figure out what's the right way to use these things, because we're kind of shooting in the dark in terms of where do you test on the plant, how frequently do you test on the plant. Right now we're just doing hole punches on leaves and hoping for the best. And the questions that are outstanding is, you need to do 10 holes, 5 holes or 1 hole to really know what's going on, and do you have to target this on like a deformation on the leaf that you think is got a higher viral load. So those sorts of studies are going on. But we're open to hear about other ones that might be out there. And of course, we can pick up SARS-CoV-2, but that's not something I think you're going to see go plant to plant. It's going to go person to plant maybe fertilizer to plant, but it's not. I don't expect that to replicate and bounce onto another plant. It's going to be, honestly, I think that's really more of a trimming issue, because I think the lights and a lot of the conditions in the grow would probably nuke that thing in a matter of days, not a week. It's really, if people are coughing on this at the counter, as you're saying, or in the trimming process, where it might get there and happen to get to a patient within a week. But the point, I think, of cleanliness in the dispensaries is probably really important. You don't want people sticking your nose or their fingers on these things at the moment, because we've got a pandemic that's currently out there. And while young people aren't necessarily affected by this, I think we really have to be cognizant of the political ramifications, because people aren't being rational right now. If they can shut down the NFL and they can shut down the music industry and they can shut down pretty much every industry at whim, I'm just baffled why we haven't been shut down yet. And I don't want to give them any reason. So while people may never use this test or use it very frequently, if this virus goes away all of a sudden, we want it as an insurance policy. So that's not the excuse. If this ever does break out and it gets blamed on a dispensary, we can be there at a moment's notice to say, there's a test for this already. We can handle this. Let's just start screening. But it's not something we want to get caught flat-footed on. If you look at what happened with COVID, reason we got so hammered with COVID here versus a lot of other countries handle it is they had their testing in order. The CDC did not have their testing in order. And I've been a pretty vocal about that and various social media platforms. So I'm the last guy who can be complaining about the CDC's lack of doing this and then just ignore doing it in our own industry. So we figured we're just going to lead by example, make a test. If it doesn't get used, who cares? It's a responsible insurance policy to do for the industry because it's probably a lethal event if it ever shows up, or it'll be a shutdown for a long period of time. And what I don't like about these shutdowns is they tend to basically you just watch Wall Street eat Main Street. I mean, all the small businesses get crushed and the big ones are the ones that are left standing at the end of them. And we're certainly seeing that in other industries. And, we've had enough of that already in the cannabis industry. We don't need an accelerant to that process.
Absolutely. So is there any other, aside from bat guano, is there any other potential sources of viruses or other pathogens as far as organic inputs? Since we're on the topic of potentially pathogenic of nutrient sources, what are the other ones maybe aside from bat guano that people should maybe look out for or avoid? Would seabird guano be equally as dangerous? Or question. What are some of the other ones out there that might be, people should use caution, or maybe they should pasteurize it themselves before using in their gardens?
I haven't seen any evidence of this being in birds yet. There's an interesting paper that looks at the spike protein and also looking at ACE receptors in a whole bunch of organisms. And of course, all the mammals have really similar ACE receptors. And so they kind of classified, whales and porpoises were really high on the list, actually. In fact, that's one theory people have kicking around, is that maybe these wet markets are actually selling whale meat or porpoise meat, because that's known to happen in some of these clandestine wet markets. But that's a more likely host for the virus than necessarily bats, if you look at the receptor stuff. But that's all, no one's seen it there. No one's seen it in pigs yet, but people are expecting to find it in pigs. So I'd say anything that might be mammalian sourced. I mean, the one thing that is known about SARS-CoV-2 is the highest viral loads are in feces right now. They persist in the GI for a long time after your symptoms clear. At least the RNA does. The people are PCR positive on feces for the longest period of time, long after it's left your nasal pharyngeal cavity. So those are the only areas that I could suspect they might be in. But again, we've not seen this. It's not been tested yet. These are all hypotheses that need to be explored. So I don't want to create any panic that no one can use any mammalian-based fertilizer because of this. We really need to just go look, because chances are these things probably are sterilized. They're probably old. I don't think the virus may not last long in the manure for all we know. So there's a lot of questions. And of course, no one's taking this stuff and putting it right in the flower, right? They're putting it in the soil at the roots. And I doubt this virus crawls its way up to a flower and gets into a growing room. So these are all lightning strike types of things that we're talking about. There's probably a bigger risk of actually human contact. And that's something that we should pay attention to in a lot of the manual steps of managing this post-curing. Curing, it's a good question, but I bet curing probably accelerates the demise of this thing. So it's really contact after curing that we should probably be keeping our eyes on.
So we had a question from chat, and this is a pretty simple question, but can insects carry viruses? And maybe the better question is, which insects are the best vector for viruses? I know leafhoppers and whitefly are probably the most notorious, but what are some of the other ones that you know of as far as insect vectors for viruses?
So I've not seen any papers on SARS getting transmitted through insects, but the lettuce sclerosis virus paper has whitefly transmission demonstrated. So that's probably the clearest cut case, and demonstrated on cannabis. I think that's where we have there's a big publication and knowledge gap is these insect studies on cannabis. Everyone's doing them on other plants, and it's not always fair to just superimpose that work on the cannabis. But hops is probably your best place to look at insect transmission of viruses in hops. It's probably the best Google area that will lead to something that might be meaningful in cannabis.
I know I've also seen evidence for leafhoppers for a beet leaf curl virus in cannabis as well. I've seen that firsthand in Oregon.
There may have been a recent paper on that in hemp. I think a few more.
Yes,
just popped
yes,
up.
there was, out of Oregon. Yep.
Yeah.
Or someone out of Oregon. Yeah. So is there any other viruses out there maybe that you're seeing or have been exposed to, but maybe you're kind of working on trying to get a profile for, or anything like that? What are some of the ones out there that you've seen? I know I've seen a wide range of different pattern mosaics, and I've seen two distinct different types of leaf curl virus, in my opinion, leaf curl virus, but very distinct, where everything else is fine except for a couple of plants that are very clearly infected with something that has this gnarly leaf curl compared to some of the others.
Yeah, so it's a really good question. I think the virus field is really at an infancy in cannabis, at least for us understanding it and molecularly characterizing it. And obviously, people have been fighting these things for a very long time in the field, but the microbes have really been where we've been putting most of our effort, mainly because they're so heavily regulated. But the viruses are also harder to go survey. So they're harder from both the technical standpoint of sequencing them, but they're also harder from collecting them out of the field in legal manners in which people can get them to our lab. RNA degrades very quickly. So asking someone to drop a sample in the mail from Oregon to us, it's not going to make it here intact. It has to be below 0.3 and all that stuff, which is probably doable nowadays, even with some leaves, but at least maybe even type 3 leaves, but or type 2 and type 3 leaves. But there's all these barriers to doing the RNA work well. So we're really trying to reach out to people who are interested in this field, who have PCR instruments closer to grows that can get access to tissues, to try to work out what are the best ways to deploy these things. Unfortunately, I only have those 4 to talk about.
So here's a question for you. So say if someone's not maybe a backyard breeder, or someone that's maybe has a facility and they're trying to get going, they're interested in this, right? They've heard this. They're really all about it. They want to go and buy a qPCR. What else do they need? Can they just take plant tissue, squish it up with whatever else comes with the tools that you have, put it into a vial, and how does this work? And do they need to have a big background, or is it pretty plug and play, or do they have to have quite a bit of training, or do they need to have a bunch of other equipment? What does this kind of stuff look like on a real world setting for people that are just maybe have no background and want to get into this?
Okay, so there's a couple different tiers. We have like kind of high-end instruments that people use in the cannabis testing laboratories that can do 96 samples at a time in 5 different colors, and those can be like 20 grand. They're real professional gear. Then we have 2 other layers of platforms we're beginning to roll out there that have varying degrees of capabilities. There's a Kai Bio station now that I think is like 8 grand, that has 16 different wells, and you can do sex testing and THC, CBD genetic testing on that thing in like 90 minutes across like 8 different samples if you do 2 different assays. Like most people, and the way we play around with that stuff is doing XY detection and then CBD typing, whether it's type 1, 2, 3, or 4. We can get off of 2 assays across 8 samples in that thing. And then you can flip that thing like every 90 minutes. It's a really slick box. It's a nice web interface. But you do need to be able to pipette. It's not hard. I can show people to do it right here. I've got a setup in my lab. These are just fancy ways of moving around. My background's not going to let you see this. Of moving around fluids. Simple pipette. If you know how to use these things, you can move 100 microliters around. Most of the assays are run off of a 4 millimeter hole punch. And I have one of those floating around here too. Now we'll do a little show and tell. So,
can you shut off your thing just so we can see your tools better, since you're
let me see if I can get that background turned
just under
on.
video, choose virtual background, and then you just hit none.
There. Get rid of that. All right.
Now we can see.
I can see. Okay. So this is a P20 pipette. The little dial here allows you to basically increment how much fluid you're going to move around. Most of the reactions we do are under 100 microliters, so you might need 2 or 3 of these pipettes that do different gauges. This handles 20 microliters. Other ones handle about 100. This is a typical 8-tube strip. You'd put 100 microliters of a lysis buffer in there, and you want to get a piece of leaf tissue about that size in there. And the way you do that is you get something from China right now. This is a grommet that you might see on your shoe, that for your shoelaces makes for a really nice disposable way of biopsying a plant to give you a perfect diameter. And it happens to fit beautifully in one of these. That's just that's kind of a hack. Wendell gets credit for this. I think Wendell's the one who's at Pheno Express, who came out with this really clever way of just sampling these things. And then usually use a toothpick or a pipette to pop the sample in there. This goes and boils for 10 minutes. And when you're done, there's enough DNA in the solution that we can pick it up with all the assays. And that's what we use to go after the RNA viruses, is what we use to go after the boil's a little bit subtly different with the RNA, just to not destroy the RNA, but it's still done in 10 minutes. And then once this is, once you have the DNA in there, you usually then pipette a little of that out with a different pipette. If you really want to go into semi-pro mode, you get one of these multi-channels, so you can do, a whole bunch in one shot. But that might be, genius level material. So once you have these in here, you move the boil into a different set of tubes that have your PCR reagents in them. And then this little device, this is an $800 device. Now, the reason this is different than the Kai Bio is there is nothing on here to read it other than your eye. So when this thing is done doing PCR, this thing needs to change color so your eye can figure out what it is and call it. And we have reagents that do that. It'll change from pink to yellow if there's a Y chromosome there, for example, or if there's a CBD gene or a THC gene. Any marker in the plant we can design an assay for that will change color on a field portable device This thing goes Bluetooth to your phone and just gives you an interface to set it up and run. These are around 800 or 900. No, this is an 8-channel one. These are 500. There's a 16-channel one that I think is like 800 or 900 now. They've put one of these on the space station. I mean, that's that light and simple to use. So all the space stations or biology is getting run off one of these things. So that's one technique we have that's kind of lower entry that gets people so they can start doing this. We've had a few grows start here, and then they walk their way up as they get more familiar. This is kind of like training wheels. The training wheels, they don't go as fast. They have error rates. They're a little bit higher error rate with this colorimetric test than what we do fluorescently. When you use the quantitative PCR machines fluorescently, the accuracy is dead on. These things, you're cutting some corners to make this happen, and the sex testing on this is probably like 98% accurate when it's like over 99.9 on the qPCR stuff. So anyway, those are some of the tools, and we're using this now to go after viruses, to go after the genetics on the plant, and eventually we're going to put a lot of the microbial tests into this format as well. So if people want to scan if they've got any microbial risk really quickly before they send it off for testing, some of these things may come into play. We know of a couple grows now that are pulling in this equipment because they're trying to just get sterile in the grows and monitor their airflow systems, monitor some of the filters they have in certain places to check on where the sources of these things are. Which is really interesting to see. The market's at this point where the grows are starting to replicate what the testing labs are measuring them by, so that they can kind of implement those same screening procedures internally, so they're not caught off guard when things fail at grows. It's kind of moving your QC internally, not necessarily to replace the lab, but just so that you're on the same kind of, grounding that they are, and you're measuring your process the same way that you're being measured in the marketplace.
So someone's just trying to get in on an entry level. What are they looking at all said and done for all the equipment, plus obviously to run it?
Yeah, we have a starter kit, which I'll probably have to post back to a group later. I don't want to misquote it, but it's probably $2,000, where it gets you all the pipettes and it gets you this stuff and a set of reagents to get started with. If you're starting off completely naked and you don't have a single pipette or a lab bench to work on. It can be a bit of a learning curve, and we work people through those things. I think if people have pipetted before and they're familiar with PCR, they usually come in at that ChI-Bio level, and that gets you off the ground under 10 grand. And those folks get a little bit more serious about throughput and are testing multiple samples for either sex or THC and CBD. As that platform gets a little bit more traction, we'll start moving some of the other tests we have on here is to look for powdery mildew and a couple other pathogens. And we'll migrate those into the ChI-Bio as more demand builds up for that. All of the assays we have on here, we also have for the adjuvant urea, which is this 96-well qPCR system. The gold standard system can run them all.
So when someone has one of these, do they have to kind of buy the software and packets, or you kind of update it as it goes once you have the unit, or how does your system work there?
Yeah, the software is we deliver it with a download that has the program that runs on here, but it's a really lightweight piece of software. And the company Mini PCR may have updates as like an iPhone app. I think I have one. See if I have the latest on my phone right here. I think I have a kind of new phone. I don't know if I updated it yet. So yeah, there's a software system here that basically runs on a phone. I'll see if I can get that in front of there. And for instance, here's it'll try and connect through Bluetooth. I don't have this guy plugged in, so it doesn't need a power source. That's one area that it's not finding the contact. But let's see, it's pretty easy to get this on there and then just edit. You can download this software on a laptop as well if you want to run off a laptop, but most people are going off this phone. And this thing just walks you through, like here's the one for CBD, kind of runs the, that's the programming conditions that run CBD. So it's kind of an interface where you just import what cycling conditions you need, and we provide those files, and it runs on Android and on OS 10 or whatever this, the Mac iOS software.
That's awesome. So do you see that being able to test for a wider and wider range of stuff in the future as far as capability? Or
Yeah, I think what you'll see over time is a lot more genetic markers pinned to the plant's genome that people want to screen for. I know people that are working on trying to figure out some of the terpenes and what assays are gonna predict the terpene profiles. I think Philippe Henry's doing a lot of that work. There's also some other people in the field that are looking at what might be governing seed sex ratio. Like, can you get? And there's some SNPs out there that may push plants to be not 50-50, but more, 75, 80, 80,
Well,
20.
isn't it? I mean, I know a lot of people have found, and myself as well, that when you breed autos, you tend to have. I've only had bred them once, but even then, we tend to have we had significantly more males than females, and I've heard that quite frequently with auto breeders. Is that?
Oh, interesting. I didn't know that. I actually read this in a patent filing from, I think it was New South Genetics, maybe John McKay's group, I think, did some work finding some markers in the cannabis genome that might predict this effect. So I don't know if he was using autoflowers or not, but that's a really interesting point. And that's another gene that we're trying to hunt down as well, is what flowering genes might be involved in autoflower. And we have a few candidates after we've just sequenced another dozen autoflower genomes. And so we're combing through those to look for copy number changes and mutations that might be governing what's going on with autoflower. So that's another marker we hope to have on the platform in the coming years. But yeah, no, I don't know what's going on there on the sex ratio thing. And likewise, Zamir Punja just put out some work where they're trying to sleuth out what's going on with hermaphroditism as well. Is there a genetic marker for the plant's potential to Hermy? That's something that's been on everyone's mind, because our current sex tests can't discern that. All Hermies come up as female. We've had a few folks tell us that there's a few Hermies they had that tested male on our UPCR assay, and we're still trying to get to the bottom of that. That was something we never expected to have happen. But so yeah, I think as more of these markers and knowledge of the genome builds, there's going to be more opportunities to put things on this platform that will, answer different questions people have about the breeding programs they have in place.
Very interesting. Oh, damn. I lost my train. Let me pull up my notes here again. So before the COVID stuff, I know you were working on a lot of stuff with canflavin A and canflavin B. Do you want to tell us a little bit more about that, and maybe a quick brief for those of the people that aren't familiar with it, what those are to begin with, and then a little bit about your work?
Oh, okay. So yeah, we were basically trying to hunt down all the genes in the cannabis genome that are involved in canflavin A and B synthesis, and we put a lot of those into our sequencing chips. The work that we're doing with Arbor and Agilent on that front they're resequencing like mini exomes that we sequence on a lot of people's samples that are behind our strain seed pipeline. We've been padding those sequencing tools with more and more genes, and so we recently added in a lot of the canflavin genes. Because we're interested in what those compounds do. I mean, there's people publishing them as being like 30 times more potent than aspirin, and maybe playing a role in cancer. And I don't believe they survive pyrolysis. They obviously you need to ingest these things differently, but we don't know what genes are governing them. But the other thing that's gotten our attention is cannabis A. This is something that popped up in the literature very recently as an interesting paper out of Vietnam. The Vietnamese government, the first thing they did when COVID started going through is they had the researchers informatically screen all the active compounds they know that exist in plants that grow in Vietnam that were theoretically active in silica against SARS-CoV-2. And so they looked for protease inhibitors. The SARS virus is really interesting in that its genome encodes one really large peptide, and then a protease comes and cleaves it into pieces. That's weird and really interesting that it does this. But as a result, the protease inhibitors have become a really important target in COVID. And they found the top of their list when they scanned something like 4,200 different compounds that they had against SARS was cannabisin A. It was the top damn hit in their scan. And so it's got everyone trying to figure out, how do we make this thing? Where the hell do you find the thing? Like, what tissue? I think it's in the seeds. And who's tracking this? Like, it's a really high molecular weight compound. It's like 594. So it falls off a lot of mass spec assays looking for cannabinoids that are like down in the 300 range. So we got to get on top of how to detect this from an analytical standpoint and see if we can maybe breed for plants that make more of it, because I think the quantities that were found in hemp were really low, like sub percentages, maybe down in parts per million, not up at like the 1% like THC or 20%, right? So I don't know that it's going to be the right, whether we have to synthesize this compound. It's a really complicated compound to make, or whether we can boost the plants or scan for plants that breed that make lots of it. I think right now just no one's looking for it, so we really haven't found any heater strains, if you will, that make cannabis in A. So we're in that molecule shares some of the same front end pipeline using the cinnamate pathway and a couple of 4CL I think is involved in there. So some of the pathway that makes the canflavins is also involved in making cannabis in A. So there's only a few papers we can find on this thing. If anyone finds papers, please share them with me, because I've only managed to find like 3 or 4 papers in cannabis in A. It's really understudied. There's probably people who have access to older journals from other countries that I don't have my fingers on. So if you find anything, I'd love to hear more about it. But I think that's an important topic. That and probably I'm kind of murder the pronunciation of this, but Q-certain is another one, or quercetin, I think is how you pronounce it. This is found in more plants than just cannabis, but this was another compound that came up as being a zinc ionophore that could work a lot like HCQ or hydroxychloroquine, and probably not have the side effects of AFib that we're seeing with HCQ, but it helps move zinc into the cell, and that's the mode of action people think's going on with chloroquine, is that it's helping facilitate zinc transmission into the cell to stall the viral replication in its RDRP gene. That if we can get that out of cannabis, that'd be fantastic. But no, I don't think anyone's tracking these compounds in the testing labs, and that would be a great project, is to find compounds in cannabis that potentially play a role in fighting off SARS. And I think the world would welcome it. There's even a preprint that's out for some groups that found some cannabis extracts recently in Canada that are fighting off SARS-CoV-2, at least in vitro, but it doesn't seem to be related to THC or CBD if you go through their paper. The news kind of bent their paper the wrong way, but they have a bunch of extracts in there, and the CBD to THC ratio has no bearing or no correlation with the activity against these viruses. So there's something else in their extracts that could be a protease inhibitor, or maybe one of these other transporters that might be playing a role. There's so much to be looked at there. So that's not, we can help on the genetic side. We can't do anything on the analytical chemistry front. So we need to partner with people in that area. But I think there's a lot of promise there that we want to keep. We want to keep those molecules on the radar so we can fully understand this plan and not be just playing this THC CBD question. There's so much more to it.
Man, that's so interesting, and it just goes to show you that there's such a deeper range of molecules that are medicinally beneficial in the plant that we haven't even remotely even attempted to test for or document or map out, or it's not even on our radar. But here we have very demonstrable results that it's actually helping with something right now. And I think that's what's so cool about this, is that we still know so little about this plant that we've dumped how many billions of dollars into researching and working with and growing over decades, and we're still completely just total infants in the knowledge of it.
So did you see that it was a hemp paper, I think it was Carmagnolia, that they found small amounts of a new cannabinoid, like maybe back in December. I think it was called cannabimavone.
It's supposed to be super potent, that one.
No, that's THCP. That's a different
THCP,
also from Italy. Yeah, we can't keep track of these things, right? They're coming out every month. Great.
Yeah, which new ultra strong cannabinoid?
Yeah. The interesting thing about THCP is the Dimitris group at CanMed presented that they found that in yeast before it was published in the plant. They noticed when they cloned THC synthase into yeast, and they noticed when they fed it like octanoic acid instead of hexanoic acid, out came THCP. I don't think they fully appreciated its potency. They just noticed that they could change the input carbon and they'd get different tails coming out of the other end. So that's kind of an interesting story on synthetic biology. But no, the cannabimavone one I've never heard of before, but I think it was actually described. Richard Rose knew about it long before this paper came out. It was described once before in hemp, and I think it got redescribed and better characterized more recently in Carmagnolia. And that's another one that we got no idea what that thing does, but I think it wins the award of having the coolest cannabinoid name. hands
So,
down.
so let me ask you a question on something that I've been working on, or at least exploring. Um, well, anyways, something I've had the pleasure of being exposed to. Maybe that's the better way of putting it. Um, so tell us more about the groups of alcohol bonded cannabinoids, because they aren't really studied much. CBT and some of those others that just aren't really talked about much. They're very hard to kind of capture and isolate and do much study with, but they are out there, and some of them have a highly medical potential for things like Crohn's disease and some others. Have you done anything with any of those
No,
alcohol
I
bonded
don't,
ones at
I'm
all?
not aware of those. Is this like CBL or did you say CBT?
CBT.
Oh, I think you mentioned this last time to me, and yeah, I was still new to me now, but no, I don't even know how those are. That's probably gonna be a question for Mark Scaldone. I don't even know how those things are made. I mean, a lot of these cannabinoids that are on the list are metabolites or oxidative byproducts, and I don't know. I don't even know where those ones fall. So I don't know a damn thing about those. Much more to learn.
Oh, that's fine. So what are some of the other areas of research that you've been focusing on, aside from your COVID work and some of the other stuff that we've talked about with flavonoids? What are some of the other areas that you're working on as far as sample tests and other things that you're currently researching, or maybe were up until COVID came to be?
Yeah, so until COVID, I was on this. We were doing a lot of methylation analysis and a lot of RNA analysis on that genome paper. So there's a Jamaican lion genome paper out there that had another 42 genomes lined up against it. We sequenced a trio, a mother, father, and the offspring of Jamaican lion, and then a bunch of other siblings, and then began to build copy number maps. I mean, the thing that's so crazy about the plant is when you look at the copy number maps, these are looking at really large sections of the genomes between the plants, and you're just seeing massive deletions and insertions all along the chromosomes. I mean, it's just one of these really plastic genomes that is very different cultivar to cultivar. You would not see this in human. This really like frightens. So I spend all my time in human biology. When I see genomes of this plastic, it just blows my mind because I'm like, oh God, we're never going to get to the end of this thing.
Like apple trees, right?
I guess, yeah. The polymorphism rate in this thing is just outstanding, and it does beg the question of what a reference genomes are, because right now, the evidence we have now is we shouldn't have one. That we really can't just take a type 2 plant and say, voila, that's the reference, map all your sequencing against that one reference, because there's probably a lot of plants out there that have genes Jamaican lion doesn't have, and that would create a bias. And so we need to create a pan genome, if you will, which is a type 1 reference, a type 2 reference, a type 3 reference, and then preferably references that have a lot of these other regions of the genome that seem to be quite disparate between the plants that we surveyed today, so that we collect a collage of all the potential genetic content that could be in a cannabis plant. Because I don't think a single plant's going to give you all of it. It might give you 90%, but there's a good 10% that's probably different amongst all the other plants out there that you need to build kind of a meta genome, if you will, or a what we call a pan genome. So we're working on that. That's one area where we're sequencing lots of other genomes to get better maps and building different references. And so we've been doing a lot of work with Hi-C and tools to build chromosomes and understand all these structural variations. And that's just to make sure we build better tools. I mean, when you don't have these references made, it's really hard to design PCR assays that can adequately tease microbes away from plants. We see a lot of entrants come into the field and try and do this, and they do it without looking at the reference, and they end up having primers that cross-react to all the wrong things. So that's one area we're working on. We've been putting a lot of effort into CanMed. We're still going to have CanMed this year, so it almost didn't happen, but it's looking like the mayor and the governor are going to allow it to happen. This is a conference down in Pasadena in late September, so they'll be doing things to spread people out and amend by all those rules. But it's looking more and more like the mayor and the governor are going to open things up by then, and that we'll be able to have conferencing happen. I think there's a limit, like under 2,000 people, and you can have the event go on. So we put some effort into that, making sure that all of those things are happening. And then we've also been updating Cannapedia. So folks haven't been there in a while. There's now tools on Cannapedia that will scan your genes of interest to look for point mutations that might be changing the way CBD synthase is behaving. I mean, that's one area that everyone wants to find a CBD synthase that doesn't leak as much. We don't have the right answer for that yet, but the way to find that is to be looking at the mutations that are in your CBD synthase genes, because there's likely to be one that someone will find that has a wider spread in the ratio of THC leaking that occurs. And so those tools are now activated for THC, CBD, and CBC synthase, and we're starting to open them up for a lot of the flowering genes. So the web reports there are becoming a little bit more 23andMe-like for each plant, where we can kind of pinpoint what gene has a mutation and which one doesn't, and a lot of discovery can be done there. So that's kind of been my focus, is just kind of pumping up those tools and making sure we can extract much more information out of everyone's sequence that's been through here.
Very cool, and it's neat to see that you know, for a very affordable price, people can actually get into this level of understanding of their own you know, plants and their own cultivars that they're working with. I was mentioning earlier about with apples. You can take every seed out of an apple tree off of a single cultivar, even from an F45, and you'll still have every single one's a different pheno. There's absolutely no difference. Almost all of your cultivars are clone only, just like bananas. Well, I don't know what the mutation rate is in bananas, but I know in apples that it's super high. But that's how Johnny Appleseed actually came to be so popular, was because he's planted. He basically did like millions and millions and millions of phenos. Searching through, and he
on Instagram. There was a great story on Instagram about kind of the history of trademark and varietals and apples, and like what we can learn about that in the cannabis field. I remember reading something about this.
No, I wasn't the one that posted it, but yeah, so that it's just, you know, especially with plants, you know, it gets a lot of plants are incredibly hard to predict, and even with all the incredible technology and all the tools that we have today, they still commercially can't get apples to stabilize, right? So and we have, you know, CRISPR and all these crazy tools that we have that we could really bend it to our will, and we still can't make it work, right? So
these plants are tricky bastards. Yeah. Well, you know, we have been, it's the cloning side of things. You know, we finally have the first glimpse of how long the telomeres are. I mean, that we didn't know that until December. So that was really fun to see. That was after December. It's actually January where that popped out. That was so Sergey Korin is a guy who wrote one of the best DNA assemblers out there called Canoe, and he recently updated it to something called Hi-Fi Canoe that handles a new flavor of sequencing from PacBio. And he got interested in the cannabis genome because now that we have these really highly accurate reads from PacBio we've never had before, the plant genomes are all buttoning up really beautifully. Like they just did the Sequoia genome, which is like I think it's like 20 gig. It's one of the biggest genomes out there. And it's been a real mess for like 5 or 6 years. We haven't been able to put this thing together. And this thing just got buttoned up beautifully once this hi-fi technology came out. And interestingly enough, it's in better shape than the cannabis genome, because the cannabis genome is more polymorphic than Sequoia and has more repeats than Sequoia. And so it's harder to assemble a genome that's like a 20th the size because of the complexity of cannabis compared to Sequoia. So a lot of the assemblers, the real experts in DNA assembly, have turned their attention now just this year on cannabis, because they realize that's the next prize. That's the next hardest agricultural genome next to Sequoia. So they've all started kind of semi-competing and playing around collegially and assembling the hi-fi data we have public on our site. And so we've got the Broad to chip in, PacBio's been chipping in, and Sergey Korin. And so Sergey's assembly came out, and it's the first time we've seen the telomeres of any significant length, and some of them are 40 kb long. I mean, these are huge. Some of them are 1,200 bases. Like, to give you perspective, like I think some human telomeres are only like 500 bases. They can vary tremendously in length, but the length of them is oftentimes reflective of your molecular age. So like Dolly the lamb's telomeres kind of disappeared and it didn't live as long. And we don't have evidence of this, but we suspect that this might be happening in clonal replication of cannabis, is that after you clone something like 50 times, its telomeres are toast and it's no longer starts having chromosomal instability. And the telomeres are important to remain stable in cannabis because most of the genetic content is out in the tips of the chromosome. It's not in the centromere. Although THC and CBD look like they're more centromeric, a lot of the other genes that we're relying on are all getting swapped and exchanged out by the tips of the chromosome. So if those telomeres erode, there's going to be hell to pay over time, which is where I think maybe, you know, some lines probably clone forever because they might have really good telomerase genetics, and other ones might be really susceptible to decay over time. We don't know these things, but we can now measure the length of telomeres in cannabis as of like probably mid-January this year. And it's kind of crazy to look at these things. I've just never seen repeat structures like that go on for 40,000 letters long of all the same 5-letter sequence over and over again. And there might be 1 or 2 subtle differences, and the sequencing now is so good that it can separate those and turn them into different chromosomes. It's like, it's mind blowing. These things in the human genome have remained, particularly the centromeres, have remained an enigma since I worked on the project 20 years ago. And only today are they like gotten across tip to tip on some chromosomes with the human. And now this new PacBio thing's out, and we're starting to be able to do this in all genomes everywhere and sort out a lot of this dark matter that's in genomes. That I don't know, maybe it's going to tell us something about cloning. Maybe we'll be stuck on cloning longer than we realize. But it sounds like from your perspective on apples, we may be on cloning for a really long time, unless we go, obviously outdoor hemp. It's not practical to hyperclone like that. You're going to go with seeds, but I haven't seen anyone pull off apomixis yet. I've not seen anyone I heard some talk about synthetic seeds, but I'm not certain that's cheaper than making clones right now. So yeah, the polymorphism problem is an issue, and we're going to get drastic phenotypes. Fortunately, I don't know if it's playing a big role in the cannabinoid content. It seems like those seem to be somewhat stable among siblings. I don't know. What's your experience there? Do you ever see like sibling plants that end up, if you've typed them all, they're all coming from type 1 parents. You tend to have all type 1 plants, but maybe on the type 3 side, the hemp farmer question is, do you see a radical spread between type 3 siblings, because they all should be type 3 offspring, in terms of whether it's bouncing above 0.3 or 0.5? I would imagine.
I haven't. I can't tell you at that just at 0.3 to 0.5. What I can tell you is, at least with my experience doing aquaponic versus soil trials over and over and over again.
There you go. Yeah.
Our terpene with the exact same cuts off the exact same moms, so that we had the same moms almost always in soil or aquaponics to start off with. So take the same cuts to make sure that the genetics are exactly identical, so there's no variation at all. On every test that we've ever done, we did none with seed, all with clone, just for the sake of uniformity. And we found CBD expression is significantly higher. THC expression on average is higher, but less so than CBD. And total terpene expression is just off the charts higher in the aquaponics, like just laughably higher.
Do you think that's is that
to me, I think it's more endophyte and secondary microbial stimulation to the immune system of the plant, triggering
acid pathway.
Yeah, so you're basically, I love to explain it to people as it's like vaccines, right? I want to give the maximum number of non-pathogenic microbe exposure to the plant's root system, so we can then know what's in the neighborhood to make things to defend itself. And by exposing it to the most number of things that are non-pathogenic, by giving it both terrestrial and aquatic microbes, it gives it the most number of reference points to make stuff and metabolite and
That's
terpene.
a good plan. I mean, I think that's what's going on with COVID right now, is that we've got populations out there that have seen some of the ancestral strains that are less virulent, and that's given them protection against the current thing that's more virulent. What you're seeing is in the most sedentary part of the population, that the nursing homes and what have you, they don't get as much cold traffic, right? The coronaviruses aren't going through there as quickly as they are going through the kindergartens, right? So all the kids are immune to this thing, or they fight it off through the innate immune system. It's either antibodies or innate immune system. They seem to be fine, and they think it's because they have prior T cell and B cells from the last year's cold, and it's giving us some partial protection against this one. Whereas as you migrate into the more sedentary part of the population, they're not seeing as much. Their contact map is not as geometric as like a kid in kindergarten, right? He probably touches stuff that like 100 other kids have touched, who've touched 100 other people. Like, their contact maps go geometric really fast. A nursing home contact map is like maybe the nurse and a couple family members, right? So those people don't build up the immune repertoire that you're talking about, and in comes something slightly more virulent, and bam, they can't fight it. So if you're priming the immune system in the plant with aquaponics, that's a grand idea. It sounds like it would probably be pumping up the terpene pathway and a lot of the other things that are connected to making cannabinoids.
Oh yeah, and we've seen that in our test results going back all the way to 2013. So, or I'm sorry, yeah, 2013. So, one other question I was really wanting to ask you, and I've been wanting to ask you this for a long time, and I think maybe I asked it before, but I don't remember, so I'm just going to ask you again. There's a lot of people that make claims about cannabis having the potential for horizontal gene transfer with nearby plants, especially in regards to terpenes. I've heard it from 3 separate people. I've never seen any evidence of it. And direct evidence, and I've heard different theories on it being microbial related or some others, but that being a theory on how, again, this is all something I'm just repeating that I've heard, that the reason why cannabis has such a wide range of cannabinoids and terpenes is because it has the ability to kind of incorporate those into itself. Is there any truth to that based on anything that you're aware of and have you found anything in your testing that supports any of that evidence wise?
So I can't say I've directly looked for that. So what I can say, kind of floating around in my dusty mind here on this, is that yeah, microbes have terpene synthase genes, but they tend to have fewer intron and exons. So they tend to be more compact genomes. So I wouldn't be surprised if those are going. There's lateral gene transfer in microbes all the time. Endophytes might have a better shot of doing that with cannabis. Now, the interesting thing about cannabis terpene synthase genes is they do have a little bit of a signature to them that would probably highlight this if it ever went plant to microbe, is that they tend to have really long introns. So the genes are, let me back up an intron and exon. Exons are, one analogy is to think about your hard drive being fragmented, where your program is actually written in little pieces spread out across the hard drive. And the more defragmented it is, that's kind of like having really big introns and genes. Introns are the regions in between the exons. The exons get excised to make the protein. So your protein could have like 10 exons, but the way they sit on the genome is they're spread out with a bunch of intergenic sequences that don't code for any amino acids, but kind of space them out for some, God knows, regulatory reason we don't yet understand. The interesting thing about cannabis terpene synthase genes is they tend to have very, very long introns, unusually long. And if we found one of those, that would be a telltale sign that maybe it moved in a different direction. And likewise, I bet the sequence alone would probably have a higher homology to the cannabis references than, let's say, if it went to hops or something. So those are I haven't looked for that. That's interesting. I wouldn't say it can't happen. And we probably have tools now to be able to hunt it down.
Well, you have the alleged story from Mr. Kyle Cushman on planting the strawberry kush in the strawberry field and getting the strawberry terpenes. So I'm just.
Oh, really? I didn't know about that. All right.
Well, again, who knows if that's really what happened or whether or not it just happened to be that those plants, because it was nearby, it happened. I don't know, some other environmental factor. Who knows, right?
Dragonfly Earth, wasn't she talking about that? Where she was claiming, I think, if I remember correctly, that when you plant with other plants, that it can, take on traits of stuff planted nearby, like flavors or smells or.
Well, I know there was someone talking, I forget who it was, one of the regenerative folks was talking about chamomile greatly increasing THC expression, which I thought was kind of an
now there's something that could be going on with the microbiome here, and that, like, if you look at some of the work in Arabidopsis and you microbial, like you put on antibiotics and you fumigate the plant with antibiotics, its terpene expression completely shifts, right? So what does that tell me is that, well, if the microbial flora in the soil is shared between plants that have the same ancestral terpene synthase kind of pathways, you might see them bend in the same direction. Like if both plants are capable of making limonene and you have a particular soil environment that promotes limonene, they might both start making it because the microbes are all communicating the same language. I would dig into David Sinclair's paper on this called Xenohormesis, Sensing the Chemical Cues of Other Species. That's a really good paper that will blow your mind on how cannabis plants, well, it wasn't about cannabis plants, but plants in general tend to be expressing certain secondary metabolites to trigger and change and alter the metabolism of the organisms when they spread their seeds. So if you look at the resveratrol pathway in grapes and in other plants, this is meant to mess with the mTOR pathway and signal caloric restrictions coming. Like we have the soils change, the environment's changed, the plant makes metabolites that tell the things that spread its seeds that you better prepare for winter here, or you better prepare for starvation or caloric restriction, because I want your body to be as properly prepared for this so my seed goes as far as possible. That's the thesis behind xenohormesis, and it's kind of a mind-blowing concept, but I think if you take that down to the microbiome level, there's probably something going on there, that the same soil environments might produce the similar bend in the terpene profile of closely related plants.
Yes,
Yeah,
that
that's
was
kind of
kind
cool
of
observation.
that was kind of my thought too. Is that there's a lot of evidence that certain microbial populations will increase certain expressions of certain terpenes. And again, if the nearby plants are having really good success with limonene defending against the local aphid population, they're going to communicate that through the microbial chain. There's heavy evidence through that, especially through mycelial networks. So it absolutely makes sense that would trigger some kind of secondary communication system to increase production of that. So,
and many parts of those pathways are conserved across plants. So I wouldn't be surprised if some of the responses, are kind of harmonized. All right, I think I've got to get some kids to bed. Actually, I didn't realize it's 11. I'm getting dinged over here.
No worries. I appreciate your time. Why don't you tell everybody how to find you and your company, and how to get one of your awesome PCRs, and how to follow you online.
Thank you so much for the time here. You can find us at Medicinal Genomics. All of the information about the different products we sell are there. We also manage the CanMed conference every year. That's try to get everyone together to have discussions like this for 3 days in a row. A lot of exciting stuff presented there, from synthetic biology all the way to analytical chemistry and what are the right growing techniques. There's a couple good kind of advancement in growing that's going to be going on there, from panels to talks this year. So that's it. I'm on Twitter, LinkedIn, and Facebook at usually Kevin_McKernan. You can find me at those 3 places. I've got an Instagram handle as well. I think there's a 73 in it though. So you can find me there.
It's Kevin_McKernan_73 for his Instagram.
There you go. Kazar 73, one of the best dead shows ever.
Thanks again for your time. Thanks again for clarifying a bunch of information. I know a lot of people have interesting ideas about the SARS-CoV stuff. I know I myself was a little bit mistaken, having misread the paper the first time. So thanks for coming on and taking the time to actually bring forth proper educational content on this issue, especially in a time of such disinformation out there.
Yeah, and thank you for making these things happen. It's important to spread the word on this. This stuff is medicine, and we got to get all the brains together to sort it out, because there's decades to centuries of information in people's heads that are not in the traditional way that we do science, and we need to interface and learn from all you guys on how to make this thing sing. And so I like that, actually. I find it to be a real challenge that we're the scientific establishment has a lot of its own problems that you can, I'm sure you guys are aware of. And so I kind of like this hybrid world of working one foot in both camps, because it's a lot more practical.
Oh yeah, and
a lot of great information. I wanted to thank you for all the great information to ponder on tonight, and also to decipher, I might say. But I wanted to say bye, since you're calling, but finally got my stuff straightened out, so I wanted to say goodbye.
Thanks again, and you're welcome back anytime. We always love the incredible amount of information that you share with us.
Yeah,
I learned so much talking to you guys, so anytime.
Thanks again, bud.
All right, take care.
That's Bye-bye. Being humble, my friend.
All righty. Well, that was an incredible episode. I think that was one of the most interesting and in-depth conversations we've had in quite a while. I know my mind is completely blown after that. That was quite a cool talk here. So big thank you again to Kevin.
All right, I gotta run. Looks like I gotta pick somebody up somewhere. All right, I didn't even know they're out and about.
Well, happy child hunting.
Yeah. Thank you, guys. Take care. Be safe.
Be safe, Kevin. Be safe, brother.
All right. Cheers.
Take care.
Cheers.
So that was incredible, and always look forward to having Kevin on, and always good times. Roger, what's new with you and your farm? I'll have a quick thing here before we switch over. We got our our brand new Aqua Punch. You can't see the label. Aqua Punch from Organic Innovations. We have it's a live diamonds, and we have quite the terp profile on, over 1% beta carpholine, almost 1% limonene. Yeah, the carpholine, limonene, myrcene, pinene are the high ones on this one, and yeah, you'll it's smooth as glass, man. I'll tell you what, it's the smoothest diamonds I've smoked in a long time. And then we also have some other great stuff. We have some Pure Kush, some GMO cookies, some wedding cake, some Bubba Kush, some Chem Dog coming out, some live diamonds. So be sure to look for the next round of concentrates out there. And then in a couple of weeks, we'll have some flower out there as well in the universe. So be sure to look for some more products aquaponically grown or organic from Organic Innovations in your favorite Oklahoma dispensary. If they do not carry our products, be sure to have them email us over at sales@organic-ok.com. Alrighty, and if you're looking for aquaponic nutrients, check out trueaquaponics.com. All right, what's up with you, Roger?
Well, we're getting, we're starting our natural farming project, and we built our cedar boxes today. We built like 4 or 5 because we got 2 different locations of the partners, and then we're gonna have a couple of gifts to other people, like-minded individuals in the area. We're gonna gift them a cedar box. We're all gonna start our IMO collections, and I'm also just receiving my dried herbs, and we're going to be starting our OHN. So I'll be in this next week or so. I should be rehydrating my dried herbs, and then we're gonna. I'm pretty sure I can get fresh ginger root, and I know I can get garlic. So I can't wait to do that, and so that's really exciting for us to do that. And other than that, we got to, as everybody knows, we got a real late start on the farm, but I've got about almost 1,000 plants of different peppers, tomatoes, a lot of really cool heirlooms, and I love peppers. So every year I buy all kind of different. I know Steve loves peppers too. We love, if you like to grow and you like to cook, peppers are fantastic, and they're just, I just love it. It's the different diversity you get in your greenhouse. So ordered a bunch of stuff from the supplier out in Colorado for our grow bags and my spray stakes and such like that. And yeah, I got a shout out to our friend Brooke Sheffield up in Asheville. He's going to help us out with some poly film and save me a few duckies, which is really awesome to, thank you, Steve, again for putting up with me on the show for 3 years. And you know what, meeting people like, everybody we meet, from Kevin to whoever, and all the friend Chris Trump, we all have Chris is so great. If you do reply to or go to reach out to Chris Trump, he'll reply to you. It's one of those people that really follow up. But a shout out to Brooke up in Asheville for helping us out. Yeah. Unless you're putting some in the mail, I don't want to see it. But yeah, anyway, yeah. So if y'all are in Nashville or Greenville or in Tennessee, look up Lotus Farm Supplies. I'm not sure if that's the exact name, but that's the farm gardens they got, and they're very knowledgeable, and they can hook you up with anything you got. So that's about it. We're just working hard, trying to keep up. We're gonna have a nice fall and winter harvest. I'm really excited about it, and yeah. Checks in the mail.
For those of you guys that can see this, this is
That's pretty, man.
It's a whole 1 gram diamond.
Wow.
I thought you guys would like to see. Kind of nice. You don't see single diamonds that big in a container very often. But the real happy with this latest batch, and man, just it comes out so terpy with the aquaponics. It's just so good. So good.
Well, we had a lively chat when I had technical difficulties.
Yeah, but I also wanted to give a big shout out to our listeners. Big thank you to everyone listening. Today we hit 8,000 subscribers on the YouTube channel, so big thank you to that. We have our little helper here, This is Gizmo, the little rescue cat we found on the street the other day. And she's been keeping everybody happy. But yeah, so thanks everybody for helping us get to 8,000. We just had our 200th episode, and man, Marty and I never thought we'd get to this point, and certainly never thought we'd be doing content as cool as the stuff we're doing tonight. So thanks everybody for listening, and thanks for all of your support over the years. Anything else new with you, Roger?
No, not pretty much. Unless I start getting into like personal life.
Sure.
Wife is Amy's doing much better and getting ready for her last little operation, and she might be able to come home here in a month or so after recovering from the final, thing. So, we made a lot of headway, in our relationship because it's been rough for the last 3 or 4 years, and I'm just happy. I'm very happy right now. I wrote 5 songs on guitar last night. I was so happy.
Sweet. Awesome. So
I didn't write words, I just wrote music.
Cool. So we have just kind of getting everything into full swing. We've been retooling some of the greenhouse, some of the space we have in there for setting up racks to do vegged out one gals to try and switch up our business model a little bit into some more mature clones. So that's working out really well, getting that geared up for that. Getting all of our 14,000 plants going. We're almost done with the irrigation on the back. We got the pumps and everything going mostly on the irrigation. We got the big 4,000 gallon premix tank up and plumbed up today. So that was pretty cool. The team's doing a great job with that. And then we're just a couple days away from finishing up, getting everything mounted, putting the drip lines in the field, connecting them up to the manifolds and going. So we're probably 7 to 12, maybe 14 days away from getting plants in the ground finally, after a whole bunch of waiting on parts and waiting on this and waiting on that and struggling with all the COVID stuff and, kind of coming back from Africa after all that stuff had kind of already kind of happened. So just trying to get that going, and then have a couple of cool new projects in the works that I can't talk about yet, but we got some really neat stuff going on. I've been busier than you can possibly believe. We're juggling all these different things right now. So lots of cool things. And then we've had a lot of people picking up with the True Aquaponic subscription service. We've had quite a few farms joining us in the last 2 months, especially with COVID and stuff. Seems like there's a lot more people that maybe had to let some people go, or maybe their employees got sick and they don't have that person they were relying on anymore for dosing their nutrients or whatnot. So definitely seeing an uptick in that. And yeah, just overall just being busy, just tons of stuff going on. And yeah, just some cool stuff in the works. And then trying to think if there's anything else I can talk about publicly right now. I don't think so. I better be careful on that one. But yeah, mostly just good things, just slowly moving forward and trying to get ready for the summer. Just getting ready to switch to starting days a lot earlier because it's too hot. And it looks like we're gonna have some another wet spell and coming up before too long. So just trying to get ready for that in regards to tractor equipment and regularly breaking parts on the tractor while we're tilling and having to repair them on the plow. And I don't know, that and just bug stuff. Have a great new guide. We we had a couple of test plants we put throughout the field just to see what bugs we're going to get out there, and one of them happened to be right next to a tree that had a bunch of woolly aphids, which they don't feed on cannabis really, but they will land on it and make it look ugly. So I did a quick how-to video on how to get rid of them today. That's quite entertaining if you haven't watched it yet. So be sure to check that out. It's short, but I guarantee you'll get a good laugh. So anyways, I think I'm just rambling at this point. Be sure to hit that like and subscribe button if you enjoy this content. Thank you everyone for your continued support. You can find this on audio format on your favorite podcast application. I posted on pretty much all of them at this point, including Spotify. And then also iTunes, Google, wherever you look for them. And then also we have them available on a video format on YouTube. and a couple of other places out there on the web. So thanks a lot for your continued support, and we'll catch you guys again on Thursday with another really awesome and informative guest. Take care, and thanks for your continued audience.